So today I will present about extracellular vesicles in ovarian cancer. And of course, tomorrow you will have a fantastic presentation about ovarian cancer, because Carlos also works in ovarian cancer. So for sure. So I will start with a little bit introduction, and you can see here, that ovarian cancer is among the different gynecological cancer, is the most lethal one, and the American Society estimates that 2,000 new estimated cases will be were in 2017, and estimated deaths were around 14 new deaths. And one of the disadvantages of ovarian cancer that is between 70 and 80% of the women which initially respond to the therapy eventually relapse and die. If we see the stage of ovarian cancer, you can see here 4 stages, where in the stage 1, the cancer is confined in one or both ovaries. In the stage 2, go to the region pelvic, but in stage 3 and 4, they spread to the abdomen or to other body parts. And the disadvantage is almost all the ovarian cancer is called the silent killer because the disease is usually detected in advanced stage, here in stage 3 and 4. So if you see the 5-year survival rate is only 20% if they are detected in this stage. So one of the challenge, of course, is try to detect in this stage, in stage 1 and stage 2, because the patient diagnosis and early stage of the disease have a 90% of 5-year survival rate. In this topic, the last year have taken an important role, the extracellular vesicles. because the extracellular vesicles have tons of information about the cell that the origin of the cells. For example, if we have a tumor cells, the information of the extracellular vesicle will be of the tumor cells. Thanks to the other speakers, they explain everything about the extracellular vesicles, so I don't need to say that the extracellular vesicle or exosomes are of 20 and 150 nanometers, etc. So thank you for that. But if we want to use extracellular vesicle as a cancer biomarker, we can isolate extracellular vesicle from different fluids like saliva, urine, and blood, breast milk, brain, cerebral, spinal fluid, amniotic fluid, semen, ascites, etc. And if we have a tumor, we can isolate extracellular vesicles from one of these fluids, and we can try to identify biomarkers from the tumors. Here we can see some candidate, protein candidate identified in exosome or extracellular vesicles, and you can see here the typical protein called CA125 that is used for ovarian cancer today, but also we can identify MUC1, MUC16, etc. But exosomes are not only biomarkers, they have a function in the system, and they can modulate different biological functions. In cancer, for example, they can modulate angiogenesis, chronic inflammation, tumor microenvironment, and resistance to cell death, for example, all the hallmark of cancer. And how, for example, they can modulate this, in the case of tumor microenvironment, for example, they can influence the crosstalk between stromal cells, the tumor microenvironment, and cancer cells. 2 weeks ago, I think, Carlos published this nice paper that they did the proteomic analysis of exosomes, reveals an association between cell-invasive and exosomal bioactivity. And they, in this work, they work with 2 cell lines called OPCAR3 and SCOP3, and the difference of these cells are the migration capacity or invasive capacity. Here you can see that the OPCAR3 in a cell migration assay are slower in comparison with the of SCOP3. So SCOP3 is a high invasive ovarian cancer cell, and OPCA3 is a low invasive ovarian cancer cell. And of course, they isolate exosomes from these cells, and they saw that OPCA3 cells secrete less exosomes in comparison with the high invasive SCOP3 cells. They did a correlation between the cell migration and the exosome, the cell migration capacity and the release of exosome, and you can see here that when you have less cell migration, you have less amount of exosomes, and when you have more cell migration, you have more quantity of exosomes. But also they did the proteomic analysis of these exosomes, and it's really simple, but they found 114 proteins, and all these exclusive in exosome derived from SCOP3 cells. And these proteins are related to positive migration of endothelial cells and mesenchymal stem cells. With this data, they did some function analysis, and what they did that was culture with different concentration of exosomal protein with placental mesenchymal stem cell or endothelial cells with both exosomes, in white with the exosome derived from OPCA3 cells, the less invasive cells, and in black, the exosome derived from SCOP3 cells, the high invasive cells. And they saw that the mesenchymal stem cells migrate more, and the endothelial cells also migrate more when they were cultured with the exosome derived from the SCOP3 cells in comparison with the OPCA3 cells. So really nice work, was published in Clinical Science 2 weeks ago. So if we think that we have the cellular heterogeneity in the tumor, that we have low invasive and high invasive, but they are not alone. There are many types of cells between a tumor. For example, we have here in green chemioresistant cells, or also we can have in red cancer stem cells. So also are a population, a small population inside of the tumor, but they maybe after cisplatin treatment, they have the capability to resist the chemotherapy, and after that produce a relapse. And when they produce the relapse, the population of these two types of cells start to increase in the tumor. So our question was, what happened with the extracellular vesicle derived from these cells, these cell types, and also what happened with the treatment with cisplatin to these exosomes? So they can have maybe a autocrine effect in the cell that in the sensitive cells, or maybe they can have an effect in a stromal cell like mesenchymal stem cell. So in my lab, we work with two models. We culture ovarian cancer stem cells we culture or enrich cancer stem cells using some methods. And we also have this model that is the cell called A2780 cis that is resistant to cisplatin, that is one of the chemotherapeutic used in ovarian cancer. So I want to show some data about the ovarian cancer stem cell field. So how we enrich ovarian cancer stem cell in the lab, we culture ovarian cancer single cell in a plate that is ultra low attachment, and we culture with some factor like EGF and FGF2, and also in hypoxia by 7 days. And after that, we got these spheres that are enriched in cancer stem cell. You can see here a picture of the spheres. And we check, for example, the typical Yamanaka genes, OX4, SOX2, and NANOC, the pluripotency genes, and we can see in the spheres, in this case, we use the SCOP3 cells, and we can see in the sphere that are enriched in cancer stem cell high expression of the three genes in comparison with the cells cultured in adherent cells, in adherent condition. Also, we measure a typical marker for cancer stem cell like CD133, and we can see here an enrichment in our sphere of 60%, around 60% of CD63 in comparison with the SCOP3 culture in adherent cells. So we have a difference there. But what we want to do is isolate extracellular vesicles. So we culture the cells, the spheres, and in this case, we also compare against adherent cells for 72 hours, and we take the conditioning medium, and we isolate by ultracentrifugation the extracellular vesicles to get a small extracellular vesicles. And so it's a population of extracellular vesicles enriched in exosomes. And after the centrifugation, we got our both groups, extracellular vesicle from the sphere or enriched in cancer stem cell, and the adherent cells. Here we have, as Carlos mentioned before, the three typical characterization of the extracellular vesicle from the sphere, and here we have the TEM, the transmission electron microscopy. We have here a couple of markers for extracellular vesicle are not present in cell lysis, like TSG101 and CD63. In the adherent cell, extracellular vesicle from the adherent cells and from the spheres. And of course, the nanotracking particle analysis that we can see here a peak in 130 nanometers of diameter. But what we want to do is see if these extracellular vesicles have some effect in another cell. So what we did was culture the extracellular vesicles with our low invasive, the same cell that Carlos used in his study, OCA3 cells. And after 16 hours, we did the scratch assay, the typical wound healing assay, and we take picture 4, 8, 24, 36, 48 hours. Also here in 24 hours, we extract RNA in other cells, of course, and we quantify the wound healing. We use this condition for the treatment, 10,000 EVs per recipient cells, and what we saw that the migration pattern in the cells treated with the extracellular vesicle derived from the cancer stem cells close the wound faster in comparison with the other group. You can see here the curve of the relative wound density and the time, and here the area under the curve of this graph that you can see here more migration in comparison with the other groups. Also, we extract RNA, as I mentioned before, and also we can see some genes related with epithelial mesenchymal transition, like vimentin, snail, fibronectin, and cadherin. And in the case of vimentin, snail, and fibronectin, they increase only in the OPCA3 cells treated with the extracellular vesicle derived from cancer stem cells in comparison with the other group. In the case of N-cadherin, in both cases, the N-cadherin increase. So we have some effect in migration, and what we some metastatic processes or related processes, and why we measure these genes and migration, because cancer stem cells also are called metastatic initiating cells. So they have some some properties related to metastasis. But as we mentioned before, we want to see also what happened with the microenvironment, and also what happened with the cisplatin. So we culture our cancer stem cells with or without cisplatin, and we, after that, 72 hours again, we took the media and ultracentrifugate and get the extracellular vesicles. So we have two groups, EVs from cancer stem cell and from cancer stem cell in response to cisplatin. And again, characterization, some markers, and the transmission electron microscopy. Here you can see the distribution size, and we don't have too much difference. You can see a little bit increase, but it's not significant in the scope in the EVs derived from cancer stem cell in response to cisplatin. But interestingly, we can see an increase in the release of extracellular vesicle from the cells in response to cisplatin in comparison with the non-treated cells. So after that, we took the extracellular vesicle and we did a similar experiment, but now we use mesenchymal stem cell because I mentioned before we also want to see what happened with the stroma, with the tumor microenvironment. And we change we check the assay, and now we use transwell to see migration. So we treat the cells 16 hours. After that, we culture in the transwell, and 16 hours after that, we measure if the cells transmigrate or not. And what we saw, you can see here, that only the mesenchymal stem cell treated with the extracellular vesicles derived from the cancer stem cell in report to cisplatin migrate more in comparison with the other group, and here is the quantification. So the good thing that extracellular vesicles derived from cancer stem cells do something, and also they do something in response to cisplatin. We have this is some data that we have in this field, but we need to do many other things. But in conclusion of this part, we can see that extracellular vesicle release from cancer stem cell induce migration and EMT transition genes in ovarian cancer cells. Cisplatin induce secretion of extracellular vesicle release from cancer stem cell, and extracellular vesicle derived from cancer stem cell in response to cisplatin induce migration on mesenchymal stem cells. So we have one undergraduate student working in this project, so I hope that she can get more data. But I mentioned that we have another model to work, that is these cells resistant to cisplatin. So we did the same that I mentioned before. We took the sensitive cells, that these cells, the C cells, is derived from these cells, the A2780 cells. We isolate extracellular vesicle with the same method from both cells, with and without cisplatin in this case, and we took 3 groups, extracellular vesicle from A2780, A2780Cs, and in response to cisplatin. The characterization again, TEM, the positive marker and the negative marker for extracellular vesicles. Also here, we can, in these cells, to difference with the cancer stem cell, we saw a little bit bigger extracellular vesicles with a peak in around 160, 150, 60. But also, like in cancer stem cells, cisplatin induced more secretion of extracellular vesicle in comparison with the non-treated cells. Now, see if these extracellular vesicles have some effect. And because these are chemo-resistant, we can maybe measure cell death. So if they have some protection in the to the recipient cells to cisplatin, so we use, in this case, the sensitive cells. We culture with the extracellular vesicle of our different group. We wait for 16 hours. We treat with 3 micromolar of cisplatin the sensitive cells treated with the exosome, and we wait for 72 hours and measure cell death. The most simple measurement, that is trypan blue, and we saw that we have a protection only. This is the basal cell death, so the cells treated without EVs but with the cisplatin, and we have a small protection, or 10% protection of only in the group of cells treated with the extracellular vesicles in response to cisplatin. But in the middle of this history, were published these 2 papers that say the same. So unlucky, but we start to see what we can do. And we start to find maybe another target, and we saw, in this case, mTOR, and as you can see here in this manuscript, mTOR fat will play an important role in cisplatin resistance in human ovarian cancer cells. And in fact, here they use the same cell than us. So maybe we can see if the mTOR is mediate the effect that we saw in the chemo-resistant cells induced by cisplatin. So what we did, our question was, mTOR could be involved in the biogenesis of extracellular vesicles released from chemo-resistant ovarian cancer cells in response to cisplatin. We did this experiment. We put rapamycin, with and without rapamycin, our chemo-resistant cells. We wait for 1 hour, and we add cisplatin. And after 72 hours, we isolate the extracellular vesicles. And you can see here the group, chemo-resistant in response to cisplatin with rapamycin, and with rapamycin and cisplatin. You can see here the activation of of mTOR in response to cisplatin. We measure this using the phosphorylation of the protein A6K. That is one of the targets of mTOR. And without cisplatin, we have a basal level of phosphorylation, but when we add cisplatin, we have more phosphorylation, so mTOR is activated. But when we add rapamycin, this activation is abolished. Remember, we saw this effect first, that cisplatin induced the secretion of extracellular vesicles in the chemioresistant cells, but when we block it, mTOR, this secretion is abolished. So now is the effect, this effect was in the secretion, so now we want to see if they have an effect in the cell death. The same strategy done before with our 4 groups. Remember that we saw a decrease in the cell death in the cells treated with the extracellular vesicles from the chemioresistant cell in response to cisplatin. But when we treat with these extracellular vesicles, this decrease is abolished. So maybe we measure with another technique, not only counting with trypan blue, but with the students sometimes then, this is blue, this is light blue. So now we want to see something more quantitative. We measure by flow cytometry using the live death staining, and we saw the same effect. And also we measure apoptosis by annexin 5-PI, and you can see here in the group treated with the extracellular vesicles in response to cisplatin, the Q3 that they are the annexin 5 positive, PI negative, so it's early apoptosis decrease, and in the group treated with rapamycin and cisplatin, this decrease is abolished. So, perfect. So we have some effects, and this effect is mediated by mTOR. So what we did after that, what we sent to Queensland to do the mass spec analysis. And I want to show of all the group with rapamycin and cisplatin, with nothing, and also with only cisplatin and the sensitive cells. And here we found, focusing in this 74 protein that are only in the extracellular vesicle derived from the chemioresistant cells in response to cisplatin, but these 74 proteins are mediated by mTOR. And when we did gene ontology analysis, we saw that 9 proteins or genes, protein in this case, are involved in DNA repair. This data I got 1 week ago, so we need to do the analysis because this proteomic was quantitative too, so we need to do a proper bioinformatic analysis. Here are the proteins related to DNA repair. So what we need to do now is see if these proteins are involved in the protection of the cells treated with the extracellular vesicles derived from these cells. We need to do many other things, but so in conclusion of this part, cisplatin-induced release of extracellular vesicle from chemioresistant ovarian cancer cells. If it's derived from chemioresistant ovarian cancer cells, decreased cells death induced by cisplatin in sensitive ovarian cancer cells. The release and effect of EVs is derived by mTOR activation. I think so. And EVs derived from chemioresistant ovarian cancer cells are enriched in protein related to DNA repair. So this data is from my student, that Albano, Felipe, and Belen. That is my group. And thanks to the university and funded Investigación. So thank you very much.
Thank you, Manuel. So, questions, comments?
Again.
I'll ask you something different. So in ovarian cancer, angiogenesis is really important for the growth. It's a huge tumors, right? So, I mean, do these exosomes do anything in
we saw in the first part with the cancer stem cell, we don't have effect in angiogenesis. We measure tube formation, the typical angiogenesis of say, and we didn't see that. And in the other one, the other experiment with the chemioresistant cells, we have, in fact, we are running today the experiment to measure tubes, but we saw in the extracellular vesicles, increase in BGF in the extracellular vesicles of the group of extracellular vesicles derived from the chemo-resistant cell in response to cisplatin. So I hope that this group increased the formation of TOP.
Just I would like to mention that we also saw some effect on endothelial cell coming from exosome from ovarian cancer as well. So, and the effect seemed to be dependent of the invasion capacity of the cell. So it means that more metastatic cell line can induce more angiogenesis. In this case, we saw, yes, we measure cell migration only, but the data suggests that potentially can change the metabolic state of endothelial cells and probably induce angiogenesis, and not only angiogenesis, also vasculogenesis as well.
Yeah, but here in the case of cancer cells that are metastatic initiating cells, I think maybe it's a dose-dependent manner, maybe with more extracellular vesicles can induce angiogenesis, I don't know, but we saw this effect in migration and in induction of epithelial-mesenchymal transition, so maybe this population of extracellular vesicles are not involved in angiogenesis, I don't know.
My question is, in fact, the first part of the question is whether you can confirm to me that cisplatin is an inhibitor of the release of nucleosides from the cancer cells, and that is one of the mechanisms by which cisplatin is stopping or is killing the cancer cell. Is that right? Yes.
Sorry, again, can you repeat the question, please?
The cisplatin gets into the cell and then blocks the release of nucleosides, like for example adenosine. It's competing with the transporter for that. And the accumulation of adenosine and metabolites derived from metabolism of adenosine intracellularly will kill the cell. So the fact that you are seeing an increase in the release of extracellular vesicles here in response to cisplatin is because of the transport of adenosine from the intracellular level, intracellular medium is being reduced, and then the extracellular level of adenosine is going down.
Yeah, really I don't know, because we don't measure adenosine. So I don't know if adenosine is released in extracellular vesicle or because there is down the adenosine. So maybe if we overexpress some in the cells, some enzyme to produce adenosine, or something. Sorry, adenosine.
Is the adenosine dependent of the expression of RAF protein, for example?
Which protein?
I don't know. RAF.
Adenosine.
The secretion of the exosome is highly dependent of the expression of RAF27A and 27B.
I don't know. Do you mean the transport or generation of?
But really, I don't know if adenosine is involved.
Just thinking that whether it's true that you have less adenosine outside because it's blocking the exit of adenosine, at least for a very short time, that could be a kind of signal to the cell to release extracellular vesicles that, if true what I show, they will generate adenosine extracellularly in a way to compensate the lack of adenosine because it didn't stop coming out.
Yeah, maybe, but really, because I don't work with it.
So you said that the accumulation of adenosine
can be in
the extracellular space can be interacting with the receptor, for example?
Yeah, it can be, but that really don't know.
Interesting. When you have these cells that are resistant to the cisplatin, and then you incubate with cisplatin, and then you isolate the exosome, right? How you know that your exosome doesn't contain cisplatin?
Yeah, I think there is a paper that said that extracellular vesicles can contain cisplatin. I don't know.
Not only cisplatin. Actually, you can charge exosome with different chemotherapy drugs, including the cisplatin, carboplatin.
Correct. So maybe the cisplatin in the doses in the cisplatin that go in the extracellular vesicle can start to induce some protection in the other cell, but yes, I was wondering because it's surprised that when you get exosome from the cell that were treated with this cisplatin, you need to measure cisplatin in the exosome. I'm pretty sure there should be cisplatin there. Yeah, for sure. And then you get this exosome and you put on other cells and you reduce apoptosis, trying to increase apoptosis. So maybe, and that is a little bit
we are, because I'm 100%, not 100%, but I'm confident that you should have some chemotherapy drug in the exosome that will be delivered to the cell.
But depend on the dose, because maybe the dose of cisplatin in the exosome can help to the recipient cells, because it's not enough to kill cells and start to induce, but it's enough to induce some DNA repair, for example, or something, I don't know. And after that, when you add the doses of cisplatin that kill the cells, the sensitive cell can be prepared, maybe, but it's only an hypothesis. I don't know if latest.
Comment, question? Students, don't have any questions?
America, please.
Comments? No? You didn't have any animal data with the exosome?
Not now. But we have a preliminary data with the same idea, and they decrease the ascites. So, but we don't have only preliminary data. So a couple of mice.
Okay, so we have 15-minute break for coffee, and then we come back for our final speaker, Christian. Okay.
Thank you.
Thank you, Manuel.